Aloha, folks. Can you guys hear me okay? All right, happy Thursday. Welcome back to the Fine and Feel here. And keeping in mind, I have 25 students in this class. But you guys are here, I'm here, we're going to get started. And we don't have too much to finish today, folks. We covered most of it on Tuesday, but there's some important information we're going to cover today. And just to remind you, that we don't have a class next week. Okay. All of the classes for Bio 130 next week, not gonna have them. I'm gonna give you guys a Thanksgiving break. So there are no classes for the lab or the lecture. Okay, just FYI. All right, let me share my screen with you. And today we're going to start by talking about nutrition and metabolism, especially going to focus on glucose metabolism. We did spend a bunch of time on Tuesday going over the anatomy of the digestive system. So we talked about the different organs, we talked about their function, what they do, why we need them. And today is going to be a little bit about nutrition and metabolism. Okay. We all have seen a cartoon or a picture similar to this before, right? That's the nutrition pyramid. And we all know that we need to eat a balanced diet that contains grains and proteins and vegetables, fruits, etc. All right. Now, if you are like me, a carnivore, now after all, I am from Brazil. We love our meat. So if you do eat animal products, that's not going to be any issue at all for you to be able to get all the essential amino acids that you need to make the proteins. But if you don't eat animal products, if you don't drink milk, if you don't eat eggs, if you don't eat any meat, then you need to be very careful in your diet in order to be able to get what we call the eight essential amino acids. Now, what do we mean by essential amino acids? Does it mean that they are more important than the other 12? No, that's not what it means. Okay. Remember that we have a total of 20 different amino acids. What are the amino acids, guys? Amino acids are the building blocks of proteins, right? We need amino acids. We're going to stick the amino acids together to get proteins. Out of this 20, 12 of them we can make. Our body has the capacity to make them. So we don't need to eat them. We're going to make them. But eight of those, and they are included right here, tryptophan, methionine, valine, threonine, phenylalanine, leucine, isoleucine, and lysine. Those here, they are called essential because our body does not make them. We need to consume them. We need to eat them. Now, if you only eat grains, like corns and other grains, you're going to be able to get the six essential amino acids right here. You're going to be missing the isoleucine and lysine. If you only eat beans and other legumes, you're going to be able to get the six essential amino acids right here from the diet. You're going to be missing tryptophan and methionine. Okay? So you need then to be able to make sure that you're eating a combination of grains and legumes in order for you to be able to get all eight essential amino acids in your diet. Because as you can imagine, folks, missing even one of those amino acids is extremely detrimental for our health. Because one single amino acid that is missing, that means several different proteins will not be able to be made because that specific amino acid is missing. Okay? Now, here we have a little table. You don't need to memorize this table. I'm not going to ask you questions about this in the exam. But it's a nice little table that you can print. You can stick it on your fridge so that you know what are some of the essential vitamins and minerals that you need in your body. Most important, what they're good for, why you need them, what happens when you don't have enough of them, how much you should be taking on a daily basis. And we're going to talk more about this in a minute. Why is this important? And then where do you get them from? What type of food items you need to eat in order to be able to get those specific vitamins. Okay? Now, hopefully, right now we are almost at the end of the semester. We only have two more chapters to cover. Hopefully, now you understand why vitamins are important to our body. You know, we have heard about vitamins since we are little kids, right? They talk about make sure you eat all your vegetables, make sure you get all your vitamins. We always knew the vitamins are important. Nobody ever told us why they are important. So hopefully now you understand, for example, that vitamin A is essential for our eyesight. If we don't have enough vitamin A, that is going to lead to night blindness. We know, for example, that vitamin B12 is essential for red blood cell formation. If you don't have enough vitamin B12, you will not be able to make enough red blood cells. You're going to feel tired, you're going to have fatigue. We know, for example, that vitamin D is extremely important for absorption of calcium. If you don't have enough vitamin D, calcium absorption is going to suffer. We know, for example, that vitamin K is very important for blood clotting. Okay, so these are the type of connections that you need to start understanding in terms of vitamins and what they do for our body. Okay, now the million-dollar question: do we need the vitamin supplementation? What do you guys think? I want to hear from you. There is no right or wrong, okay? So you can feel free to speak up.
I mean, depends on your diet, right? If you have a very balanced diet, you probably would need less to none. But if you have a poor diet in terms of nutrients, you probably want some supplementation.
Absolutely. Now I have a further question. Raise your hand if you think that you have a balanced diet on a regular base. Right? So pretty much that applies to most of us. So I do think, and again, don't take medical advice from me. I am a fake doctor. I have a PhD, not an MD. But from what my research, from what I understand in terms of health, I do think vitamin supplementation is important for the general population. Because again, let's face it, let's put the mirror in front of our face. Most of us do not have a balanced diet on a regular base. Okay. And if there is vitamin supplementation, then again, that is great. Now, this is especially important for mothers that are expecting, that are pregnant, for the elderly. And I want to say something else, though, about vitamin supplementation. Not all vitamin supplements are created the same. Okay. You know, if you go to Costco. I don't know how many of you shop at Costco. I do. I love Costco. It's like therapy for me, going shopping. But if you ever been to Costco or any other, grocery store or Long's Drugs or what have you, you know there's an entire aisle of vitamin supplementation. Okay? Now, the thing is, though, not all vitamin supplements are actually absorbed by the body. And that is very important. Look, some people think they are, you know what? I take my vitamin supplements, I pop that pill every morning, I am covered. Not so fast. We need to know if that vitamin supplementation is actually absorbed by our body, because a lot of them are not. And they're not going to tell you on the bottle that this supplement will not be absorbed by your body. Therefore, you're going to be pooping it out. Nobody's going to say that because the supplement business is a huge, massive business. Anybody can put anything in a bottle and say that is a supplement, and that's it. There is no regulation whatsoever of a supplementation. Zero. If you're going to put a drug on the market, you have to go through several hoops. You have to go through several clinical trials. It's extremely expensive. It needs to be regulated by the FDA. Vitamin supplementation has zero regulation. Okay? Now, the reputable business, they will test the supplement for absorption. And it's going to say on the bottle, this has been tested for absorption. That's a good thing. It's a good beginning. Okay. So pay attention on all those different vitamin supplements out there. Not all of them are actually effective at all because they're not absorbed. Now, let's talk about something called hypervitaminosis, which can be a serious issue. And hypervitaminosis means too much intake of vitamin. Some people have this idea, guys, this thought process. If my bottle of vitamin says that I should take one pill a day because that is good for me, I'm going to take 10 pills a day, which is going to be 10 times better for me. Does that make sense? Is that a correlation there? No, it does not. Okay? In fact, taking too much vitamin is actually detrimental for your health. Not only going to be wasting your money because you're going to be taking more than you require, but it also can be detrimental for your health. It can actually get you sick. Let me give you one example. Over in consumption of vitamin A. Okay, excess intake of vitamin A has been shown in several different studies to increase osteoporosis by inhibiting osteoblast activity, and especially problematic for the older folks, right? A lot of people, they already have the tendency to have osteoporosis, especially women in a menopause age. And if they are intaking excess vitamin A, that is going to exacerbate the problem, right? Any questions, folks? All right. Now, at the end of the day, why do we eat? Why do we take vitamin supplementation? Where does all the nutrients go? They go inside of ourselves. Of course, we like the taste of food. We take pleasure from eating. And that's something that we evolve. This is a process of evolution. The reason why we like certain things, we like to eat them. Now, at the end of the day, the whole point of eating and consuming all of those vitamins is so that we can keep our cells functioning, keep them alive, keeping them dividing, keeping them healthy and functioning. Okay? Now, let's talk about metabolic reaction and metabolism. And before we go any further, let's define what metabolism is. And a lot of people are confused about the word metabolism. Guys, metabolism are all the chemical reactions happening inside of a cell. Okay? All possible chemical reactions happening, going on inside of a cell, they are grouped together as cellular metabolism, right? Now, if I could take a picture of all the chemical reactions happening in a cell at a specific moment in time, this is what the picture would look like. Right? So we're looking here in this graph, this crazy looking graph, the metabolism of a typical cell. Right? All the pathways, all the chemical reactions that is happening within the cell. Now, are we going to talk about all of these? Absolutely not. We're going to focus on this little yellow shaded area right here, this area, and this area is showing glucose metabolism. And that's what we're going to talk about right now. Now, we're going to go in a lot of depth about it. No, we're going to keep it at the surface, but I want you guys to understand how important glucose is for our cell activity, okay? Now, we do have two different types of metabolism or two different types of metabolic reactions. We have anabolism and we have catabolism. What is the difference between the two, folks? In a catabolic reaction, we're going to take a large molecule and we're going to break it down into smaller ones. Let me give you an example. When we take a protein and we break down the protein into amino acids, we are doing a catabolic reaction. When we take DNA and we break it down into nucleic acid, we are doing catabolic reaction. Anabolic reaction or anabolism is the opposite. In an anabolic reaction, we are taking small units and then we are building something bigger and more complex. Okay. Now, what all of these here they have in common? Carbohydrates, fats, proteins, all of them, what they have in common in the context of our cells is that they are all fuel sources for the cell. All right? Out of these three right here, guys, without a doubt, glucose is by far the most important, the number one fuel for our cells. And this is why, and I mentioned this before, we should not completely remove carbohydrates from our diets. Okay, because carbohydrates are packed with glucose. We get a lot of glucose from carbohydrates. Now, yes, we can do gluconeogenesis, and gluconeogenesis is the formation of glucose not from carbohydrates, but from fats and proteins. Remember, we watched the video of that doctor, the lady, talking about type 2 diabetes and how to reverse type 2 diabetes. She was a little too extreme, in my opinion, and also YouTube's opinion, because there's a warning on that talk. But she says that, you know what, we don't need carbohydrates at all. And that is not true. Now, the problem, though, is that our diets, the diet of today, is heavy on carbohydrates. If you go and you buy, a plate lunch somewhere, you're going to have this mountain of rice and a little bit of vegetable and a little bit of protein, right? It's too carbohydrate heavy. That is the issue. It's not to remove the carbohydrate completely, but to decrease the consumption of carbohydrates. Okay? Now, when we talk about fuel for the cell, what are we talking about here? We are talking about the synthesis of ATP. So all of those, glucose, fat, proteins, they are all going to be used to synthesize ATP. And we know that ATP is the energy currency of the cell. Okay? Now, let's talk a little bit more about glucose and see what happens. Why do we need this glucose? What's going to happen to it? Where does it go? Why we need to take it? So here we are looking at a mitochondrion, right? And we know that the mitochondrion is singular, mitochondria is plural. It is what we call the powerhouse of the cell. Okay, we know that most of the ATP will be produced by the mitochondria. Now, pay attention. I didn't say all of the ATP. I said most of the ATP that a cell needs, because there are some ATPs which are going to be made outside of the mitochondria. Okay, so now here we have glucose enter the cell, and now this glucose, which is still in the cytosol of the cell, is going to undergo a process called glycolysis. Now, what does glycolysis mean? Glyco means glucose. Lysis means to break down. So this is the breakdown of glucose. So when glycolysis is going to break down glucose, glucose is going to go from a 6-carbon ring to 2 3-carbon rings called pyruvic acid or pyruvate. Now we can see right here during this process, we get some ATP, a couple of ATPs right here when we go from glucose to pyruvic acid, but it is very little. Those ATPs that are produced when we break down glucose into pyruvate, they are what we call the substrate level phosphorylation or SLP. All right, so SLP is going to give us a couple of ATPs. Is it enough to sustain a cell? Absolutely not. Now, the pyruvate that was formed from the breakdown of the glucose, they are going to enter the mitochondria right here. And now this pyruvic acid or the pyruvate, they're going to go through different chemical pathways, different chemical reactions. One of them is a chemical reaction called the Krebs cycle. And you don't need to worry about this. Don't need to memorize this. What I do want you guys to understand is to understand what happens, what is the result of this pyruvic acid going inside the mitochondria and then going through those chemical pathways. And the result is the formation of what we call electron carriers. And we have two different types of electron carriers. We have the NADH and we have the FADH2. Both of them are electron carriers. What else we're going to produce when we break down this pyruvic acid? We are going to generate electrons and we are going to form CO2 right here. So guys, now you know where that CO2 is coming from, the CO2 that we exhale. The CO2 is going to be formed when we break down the pyruvate by the mitochondria. It is a byproduct. It is a waste product of the process of breaking down that pyruvic acid. Now, what about this electron carrier and the electrons right here? Guys, these electrons, they need to be moved to something called an ETC. What is ETC? ETC stands for electron transport chain. And those electron transport chains, as the name implies, they need a source of electrons in order to be able to move this electron around and then eventually generate ATP. We're going to see how this happens in the next slide. Now, these electrons here, they need to be delivered to the ETC. Guess who will deliver the electrons to the ETC? NADH and FADH2, the electron carriers. I want you guys to think of NADH and FADH2 as little Ubers. They're going to grab the electrons and going to drive those electrons to the ETC. What is the ETC after all? The ETC is a bunch of enzymes that are embedded in the inner mitochondrial membrane. And again, we're going to see this better in the next slide, okay? But these are enzymes embedded in the inner mitochondrial membrane. Now, as these enzymes take those electrons and move them around from enzyme to enzyme, we are going to accumulate hydrogen ions in this space right here into what we call the intermembrane space, which is the space between the outer mitochondrial membrane and the inner mitochondrial membrane. Okay? And also, we are going to be using oxygen gas, O2, and here we go. O2, oxygen gas. This is why we inhale it. We're going to need this oxygen gas because the oxygen gas is what we call the final electron acceptor in the ETC. It's going to be the final place for the electron to move. Because if we remove the oxygen gas, ATP and the entire ETC electron transport chain is going to stop. There will be no ATP. Okay? If we remove glucose, what's going to happen? You're not going to form NADH, FADH2. You're not going to form electrons to drive the electron transport chain. So we need the glucose from the food that we eat. We need the oxygen gas from the air that we breathe. We know that if we stop eating, eventually we're going to die. If you stop breathing, eventually you're going to die. Why? Because you will not generate ATP. Okay, so now let's do this. Let's take, by the way, this process of forming this 34 ATPs here. This is what we call oxidative phosphorylation. So we have oxidative phosphorylation and we have a substrate level phosphorylation. We can see that oxidative phosphorylation is much more efficient. We can get 34 ATPs that way, but it does involve oxygen gas. Substrate level phosphorylation, it does not involve oxygen gas. All right. So let's take a little slice of the inner mitochondrial membrane right here. And let's amplify this. Let's put this under the microscope, okay? And I want everybody's attention here. This is something that a lot of people go through biology, go through chemistry, and never learn. Let's understand this once and for all. It's not that complex. At least let's understand the big idea, the general concept. All right? So we have this little slice of the inner mitochondrial membrane, and we're going to amplify this. So here we can see this is the inner mitochondrial membrane, which, by the way, is made of phospholipid bilayer, just like the cell membrane. And here we have the enzymes of the ETC. Okay, you don't have to memorize any of this right here, but these are the different enzymes of the electron transport chain. Okay, now here we are. We have the NADH, the little electron carrier, the little Uber for electrons, is going to drive all the way to the ETC and is going to deliver an electron to the enzyme right here. Here's the electron. Now, this electron, let's follow it. This electron is going to move from enzyme to enzyme to enzyme to enzyme to enzyme. Every time that this electron moves from one enzyme to another, folks, we are going to have the movement of hydrogen ions into the intermembrane space of the mitochondria. So the movement of electron drives the pushing, the transport of hydrogen ions from the inner section of the mitochondria to the intermitochondrial membrane, right here. Every time this electron is moved. So what's going to happen after a while, folks? We're going to have a huge concentration of hydrogen ions in the intermembrane space. This is what we call a chemiosmotic gradient. So we have this huge chemiosmotic concentration of hydrogen ions in the intermembrane space. Now, I have a question for you. We know that when we have all these hydrogen ions right here and very little hydrogen ions over here, this system is not in balance. This is not homeostasis. Homeostasis means balance. This is not imbalance. The real question then is, and I want everybody's attention here, please. You know the answer to this. Can the hydrogen ion that are in the intermembrane space move back across the membrane through simple diffusion? Yes or no? They want to move across. Can they do that through simple diffusion, folks? Again, the question is, can the hydrogen ions right here move back across the membrane through simple diffusion? You know what simple diffusion is, right? We talked about it. Is the movement straight through the phospholipid bilayer. Can the hydrogen ions do that? Yes or no? Come on, guys, make me proud. Okay, I'm going to give you a clue, okay? Can the charged hydrogen ions move across the phospholipid bilayer directly through simple diffusion?
No.
No, thank you. Philip saved all of you guys. It cannot. I don't know if you flip a coin or not, but it doesn't matter. It's the right answer. Look, it cannot. Why not? Because charged ions cannot go through the phospholipid bilayer directly. Why not? Because of the fatty acid tails in the phospholipid bilayer, and they will repel anything that is charged. We talked about this when we discussed diffusion. Okay, charged ions in large molecules cannot move through simple diffusion. Okay, so how is the hydrogen ion then going to move? The hydrogen ion will need a transmembrane protein right here. And this transmembrane protein has a name. This is what we call ATP synthase or ATPase. Now, ATPases not only let hydrogen ions move across, they transport hydrogen ions, but every time that a hydrogen ion moves across, it gives energy to the ATPase to synthesize ATP. Another hydrogen ion moves across, another ATP is going to be synthesized. Okay? Now, what's going to happen to that hydrogen ion once it goes through the ATP synthase? That hydrogen ion is going to combine with oxygen gas and combine with the electron here that is moving and is going to bind to the oxygen gas, and together, the plus hydrogen ion, and together they are going to form water. Water is a byproduct of oxidative phosphorylation. Okay, it's a byproduct. And this is why, guys, we're going to talk about the urinary system in the next chapter. What do we do? What is the last thing that we do before we go to bed at night? We go to the bathroom and pee. What is the first thing that we need to do when we wake up in the morning? We go to the bathroom and pee. Now, I don't know if you're waking up in the middle of the night to drink water. Most of us are not. So where's all the water coming from that we have to pee it out? Here. That's where the water is coming from. Okay, it is being formed during the process of aerobic respiration, during the process of um, phosphorylated oxidation. Okay, now, and this is why we need the oxygen gas, because again, I'm going to repeat that. Oxygen gas is what we call the final electron acceptor. This electron right here that is moving from enzyme to enzyme in the ETC needs to find the final resting place. And the final resting place of this electron is oxygen gas, which again will combine with hydrogen ions to form water. Okay? So we're going to end up with water and ATP. That is the end result of oxidative phosphorylation. What happens if we don't have oxygen gas, guys? If we don't have oxygen gas, the entire electron transport chain will stop. If the electron transport chain stops, the movement of hydrogen ion across the membrane will stop. If there are no hydrogen ions, ATP synthase cannot generate ATP because it needs the power from the movement of the hydrogen ions across in order to synthesize ATP. Guys, any questions? Nothing. All right. Excuse me. All right, let's move on here and change gears a little bit. Let's talk about the role of liver in metabolism. I hope now you guys have a better appreciation of our liver, right? You know, we all think about how important our heart is, how important our lungs are, how important our brain is. Our liver does a lot. We have already talked about the liver before. We know, for example, that the liver is going to synthesize bile, right? The bile is important for the breakdown of fats. We know that the liver also produces cholesterol. 80% of the cholesterol that we need in a day comes from the liver, which means that if you remove all of the cholesterol from your diet, you're only going to reduce your total cholesterol by 20%. That's it. It's not going to reduce any further. So now, doctors and nutritionists and dietitians, they are understanding that diet cholesterol, it's not that crucial for our cardiovascular health. We used to think there was a lot of studies out there before, and this is old study, debunked study, that says, for example, that eating egg is bad for you because there's a high cholesterol content in eggs. Now, new studies have shown that that is not the case because dietary cholesterol does not make a significant impact on our cardiovascular health. Okay? So now the focus really is on the endogenous cholesterol that is made by the liver. That's much, much a significant amount. All right? Now, another important function of the liver is for the storage of glucose. We also have talked about this before. We know that the excess glucose will become a glycogen and inside of the liver and be stored inside of the liver. The process of making glycogen is something that we call glycogenesis, and it happens inside of the liver. So glycogenesis is the synthesis of glycogen from excess glucose. And this glycogen is stored in the liver and our muscles. Now, what happens when our blood glucose level is low? Now we're going to have the release of glucagon by the pancreas, right? And then glucagon is going to go to the liver and the muscles and say, hey, break down that glycogen, release the glucose. And the process of breaking down the glycogen is what we call glycogenolysis. Okay, the lysis, the breakdown of glycogen. Now, there's another important process that also happens in the liver, and that is something that is called the gluconeogenesis. And I spoke about it a few minutes ago. What is gluconeogenesis? Gluconeogenesis is the synthesis of glucose, not from carbohydrate, but from fats and protein. Now, unlike the lady, the MD that gave the talk about type 2 diabetes, talking about no, we don't need the glucose because our body can do gluconeogenesis and produce glucose. Yes, it can, but the amount of glucose that is made from gluconeogenesis is not enough to sustain our body on a daily basis. It's not enough. This is why we need the carbohydrates to get readily available source of glucose, even though our body does have the capacity to make some from fats and proteins. Now, we're also going to talk about the fact that breaking down fat to get glucose is also problematic. And we're going to see this in a minute, right? Now, let's talk more about cholesterol and something called lipoprotein. And I want to make sure that you guys understand the difference between the two because there's a lot of confusion in people, in students, in which they group cholesterol and lipoproteins as if they were one, and they are not. Cholesterol is a lipid, is a fat, is a type of fat, and is a fat that is synthesized by the liver. And we need the cholesterol in our body for several different reasons, including to make steroid hormones like testosterone, like estrogen. We need cholesterol to make vitamin D. We need cholesterol to make bile. We need cholesterol to maintain the flexibility of our cell membrane. So the cell membrane is not rigid and it doesn't break apart to actually fluid. And is the cholesterol that allows the fluidity of the plasma membrane. Now, here's the deal though. Cholesterol is a fat, and because it is a fat, it's not soluble in water. It doesn't dissolve in water. So the body has a problem transporting cholesterol and other fats in our blood. Because again, they do not diffuse in water. They don't dissolve in water. So to solve the problem, we have something called lipoproteins. Lipoproteins are carriers of cholesterol and fat. Lipoproteins will take cholesterol and other fats and transport them in our blood by encapsulating them into this little molecule made of fat and proteins, hence the name lipoproteins. And we have four different types of lipoproteins. We have what we call the chylomicrons, and the chylomicrons are lipoproteins that transport dietary fat, dietary cholesterol, the ones that we eat, that we consume. We have high density lipoprotein, low density lipoproteins, and very low density lipoproteins. High density lipoproteins, or HDL, guys, they are what we call our good lipoproteins. Why are they called good? They are called good lipoproteins because they're going to take cholesterol from the tissue and send the cholesterol to the liver so the liver can get rid of the cholesterol as bile. So basically, HDL helps with the removal of cholesterol from the body by transporting the cholesterol to the liver. LDL is the bad lipoprotein because it does the opposite movement. LDL will take cholesterol from the liver and then send this cholesterol and deposit them in the tissues and in the cells so that they can be used. Now, very low density lipoproteins, VLDL, are also called bad lipoproteins because they act just like the LDLs. In fact, VLDLs will transform into LDL as they are moving through our tissue, especially our muscle. Okay? So understand the difference between the two here. Now, why do we say good and bad? It's not really a great term because we need both of them. Each one of them has a function. Now, the real issue here is the ratio of HDL, LDL, and VLDL. As you can imagine, if you have too much LDL and VLDL, that's going to be bad. Why? Because we have a lot of cholesterol being deposited into the tissue. The more LDL and VLDLs you have, the more cholesterol is going to be dumped into the tissue. And that can lead to the formation of plaques in the cardiovascular system. If you have too little HDL, is also problematic because that means you don't have enough cholesterol being removed from your body. Okay, it's not that you only need one or the other. No, we need all of them, but we need to have the right ratio of each one. We should have a total cholesterol of under 200 milligrams per deciliter of blood. LDL should be under 100 milligrams per deciliter of blood. HDL should be over 50 milligrams per deciliter of blood in women and men, and over 60 in women. Okay, and VLDL should be under 30 milligrams per deciliter of blood. If you ever done a lipid panel, which is a blood test that you can do, all of you can do. I'm sure you're too young to do that. You don't have to do it yet. But if you ever done one, this is what they're going to do. They're going to take your blood and then they're going to look at the amount of total cholesterol, LDL, HDL, and VLDL to make sure that they are within the healthy range. Any questions, guys? All right. So again, I just wanted to put this slide right here just to point out the differences between cholesterol and lipoproteins so that you never have to think about this again. Never confuse the two again. Cholesterol is a lipid, is a fat. It is essential for cell membrane, making hormones, vitamin C, and bile, and is not water soluble, okay, which means they cannot dissolve in water. Now, lipoproteins are transport particles made of proteins and fats, so lipids and proteins. And what they're going to do, they're going to take the little lipids, the little cholesterol, triglycerides, the fats, and they're going to pack them in that little bubble. and then transport them in our body. Okay? They are carriers of cholesterol. So let's watch this video here that talks a little bit more about cholesterol metabolism.
Opioid addiction gets between you and what you love. Once monthly.
Despite having a bad reputation as a high risk factor for cardiovascular diseases, cholesterol is an essential component of all animal cells. It is an integral part of the cell membrane, providing membrane fluidity and participating in a number of cellular processes. Cholesterol also serves as a precursor for production of bile, steroid hormones, and vitamin D. While the body can obtain cholesterol from food, many cells synthesize their own endogenous cholesterol. Cellular production of cholesterol is under negative feedback control. Low levels of intracellular cholesterol induce its own production, while high cholesterol levels inhibit it. Cholesterol, together with other lipids, is transported in blood plasma within large particles known as lipoproteins. A lipoprotein is an assembly of lipids and proteins. Lipoproteins are classified based on their density. Because lipids are lighter than proteins, particles that contain more lipids are larger in size but have a lower density. Different types of lipoproteins have different sets of proteins on their surface. These proteins serve as address tags determining the destination and hence function of each lipoprotein. For example, low-density lipoprotein, LDL, carries cholesterol from the liver to other tissues, while high-density lipoprotein, HDL, returns excess cholesterol to the liver. Major events in cholesterol metabolism include dietary cholesterol is absorbed in the intestine and carried via blood circulation to the liver. The liver packages its cholesterol pool, a combination of endogenous and dietary, together with triglycerides, another type of lipid, into particles of very low-density lipoprotein, VLDL. VLDL travels in the bloodstream to other organs. During circulation, muscle and adipose tissues extract triglycerides from VLDL, turning it into low-density lipoprotein, LDL. Peripheral cells take up LDL by endocytosis using LDL receptors. Cholesterol is used in cell membrane and other functions. Excess cholesterol is exported from the cells and delivered to high-density lipoprotein, HDL, to be returned to the liver in a process called reverse cholesterol transport. The liver uses cholesterol to produce bile. Bile is secreted to the intestine, where it helps break down fats. Part of this bile is excreted in feces. The rest is recycled back to the liver. LDL has the highest cholesterol content and is the major carrier of cholesterol in the blood. High levels of LDL in the blood are associated with cholesterol plaque buildup and cardiovascular diseases such as heart attacks and strokes. For this reason, LDL is known as bad cholesterol. On the other hand, HDL is called good cholesterol because it removes excess cholesterol from tissues and the bloodstream. Common drugs used to lower cholesterol include inhibitors of endogenous cholesterol production, inhibitors of intestinal cholesterol absorption, and inhibitors of bile reuptake.
Okay, guys, any questions? All right. A little bit about lipid metabolism, and we were talking about earlier about gluconeogenesis, right? And we understand that gluconeogenesis is the generation of glucose not from carbohydrates, but from the breakdown of fats and proteins. And besides the fact that the generation of glucose from gluconeogenesis is not enough to sustain us, there's another added factor here. And that is the fact that when we use fats and we break down fat to generate energy for the cell because you're not getting enough glucose, let's say you are on one of those glucose, restricted diets, and what's going to happen is that as the fat is broken down through a process called ketogenesis, there's going to be generation of this molecule here called ketone bodies. Why are ketone bodies bad? Ketone bodies are bad because they are very acidic. They are going to cause a condition as they accumulate in our body, in our blood. They are going to cause a condition called ketoacidosis, in which the pH of our blood is going to drop dramatically. And by now, you understand that a significant shift in the blood pH is very detrimental. It will kill a person, it will kill an individual. The pH needs to be a very narrow range between 7.35 and 7.45 in the pH scale, slightly alkaline. Okay? And this increase in ketone bodies will change the pH dramatically, will become more acidic. And this is something that is very common during starvation, if people are starving, or during individuals that are diabetic. Why? Well, because if you are diabetic, you're not getting the glucose into your cells. So your cells need to use different fuel source to generate ATP. And it's going to use instead fat, lipids, and break them down, and in the process, release ketone bodies. Okay? And finally, guys, last thing I want to mention about a specific metabolic disease called the PKU or phenylketonuria. Now, I don't know if any of you have kids out there. If you do have a kid, you might have noticed that as soon as the baby is born, the nurse or the doctors, they're going to take the baby, they're going to prick the baby's foot, and then it's going to collect a few drops of blood from that baby. They're going to put into a card that'll kind of look like this. What are they doing? Well, are they doing that to the poor little baby? They are testing the baby for different genetic conditions. Among them, they are testing for PKU. What is PKU? PKU is a genetic disorder, is a genetic condition in which the gene involved in the production of an enzyme called phenylalanine hydroxylase, that gene is not functional, which means that that individual is not making phenylalanine hydroxylase enzyme. What is the job of phenylalanine hydroxylase? Phenylalanine hydroxylase, guys, is going to take an amino acid called phenylalanine and transform it into another amino acid called tyrosine. That is the normal process. All of us, I'm assuming none of us have PKU, all of us can do this. We have the phenylalanine hydroxylase, which is transforming phenylalanine into tyrosine. People that have PKU, they don't have that enzyme. What does that mean? It means that there is going to be an increased level of phenylalanine in the body. And that increased level in phenylalanine is very, very detrimental for the baby's health. That is going to lead to brain damage, intellectual disabilities, behavioral issues, and seizures in that little baby. Now, how do we treat? There's no treatment, it's not going to cure it, but how do we manage PKU? First of all, it's something that is going to be managed for the entire duration of that individual's life. Now, why do we need to know this so early on, as soon as the baby is born? Because if a baby does have PKU, that baby cannot consume aspartame, and aspartame is a artificial sweetener, and that baby cannot consume animal proteins. So you don't want the baby to be breastfeeding in the mom, because that is animal protein. Okay, because immediately if that baby has PKU and starts consuming animal protein, phenylalanine will jump up dramatically, and all of these issues here will happen. And they are permanent. Once you have brain damage, that's permanent. not gonna, come back from that. But if you do find out that they do have PKU, now the diet that's going to be managed through the diet. You got to avoid aspartame. You got to avoid any animal protein. You got to consume plant-based proteins only. And again, that is going to be a lifelong situation. Okay? Any questions, folks? Nothing.
What
All right.
percentage of children have that?
Good questions. It's rare. It's very rare. I don't know the exact numbers of how many kids per thousand kids born have it, but I know it's extremely rare. It's not very common. Okay, but is deadly and is um, very damaging for the baby. So that's why it became a world, used process now that they immediately going to test the baby for PKU as soon as they are born. Any other questions, guys?
Do we need to know the baby teeth as well as the wisdom teeth, or just the
Just the adult.
not the wisdom teeth, I mean the adult teeth as well as the baby teeth?
Only the adult. Yeah.
Thank you.
Mhm. Anything else, folks? All right, guys. The quiz for chapter 14 is open right now. You have until Sunday night, 11 p.m. to take the quiz. Again, no classes next week. Have a great rest of your week, a great Thanksgiving week, and I'll see you guys after that. Have a good one, folks.
Thank you.
Thank you. Happy holidays.